So today, given the theme of the workshop, I thought we'll talk about crop improvement in maize in a changing climate. It was a privilege to talk after May. She set up the tone and the bigger picture very nicely, and it was a colleague of mine, and really nice to see her given great talk. So the first question, and you will uh, uh, get to why we are emphasizing this question of how genetic gain contribute to improvement, is a tremendous successful story in temperate maize, and this is because of a germplasm developed at Corteva over about 100 years. You can see the increases. It was about three times at least, depending on how you want to see it. And there were many different reasons throughout this trajectory that led to the increase in production. The first one was about double crosses, and hopefully you realize or see the interaction of the connection between the breeding objective and the technology that was developed at the time. Double crosses were main objective to keep plants standing so farmers can fully benefit from mechanization. The next wave was a transition from double crosses to single cross hybrids, and the main breeding objective here was realizing the uh, transla- transformation of the resources, water, fertilizer into yield. Uh, the next wave came with the biotech and prediction capabilities. And really, finally, and it was a privilege to be part of the team that delivered Aquamax in Brazil, is this the development of drought tolerant maize and, of course, other resources that start coming to deliver on circularity and create the technologies that will bring to life many of the ambitions and the words that are written at United Nations SDGs. So this is a pretty compelling evidence. There was about 20,000 records that went into the creation of this figure here on experiments of the many years, and we'll get back to that. The problem with this approach to breeding is that the food system that we created, it's generating about twice the problems in terms of human health and environmental externalities as the solution is providing. It's about $1 trillion expenditures in food in just the US alone, but it costs about 2.1 trillion in the healthcare due to non-reportable diseases, cancers, and obesity. So we need to rethink how we do breeding. And part of the story was the short-term uh, objectives sort of blind us from taking that longer look that is required to prevent some of these problems. And circularity is a very attractive paradigm. Of course, is a metaphor. Horticultural systems are open systems, but nonetheless, it's still looking at designing waste, recycling materials, nitrogen, regenerating resources, and create value for growers and society. So, how are you going to create a breeding framework that is going to enable that? And we can build upon the quantitative genetics foundation that it lead us to the creation of Aquamax in Corteva. So this multidimensional framework I'm alluding to that is going to be required to solve part of the problems that we are creating, of course, unintentionally, is to empower decision makers, in this case breeders, to approach dilemmas. They are not going to be a free lunch as we start thinking in trading water for carbon and crop yields. Of course, changes in land use and where we are going to grow crops is another part of the story. So today, think about these multiple trades and the crop figure on the lower left of that circle, breeders just focus on agronomics, morphology, quality, yield. However, they, for some of the reasons that Mai talked before, despite the great advances in phenomics, some of these require longer term simulations. For example, dealing with carbon sequestration. If you're going to breed for improved carbon sequestration, it's going to take a decade to realize or be able to measure any improvement in any change of the system for any particular crop. Therefore, we need to start thinking a whole different prediction framework that enable us to think in these long-term consequences at the time that we can make decisions for the short-term production of food. And the same thing, it goes for water quality and use efficiencies. It's also any design framework that enable us to transition from linear to circular economies and certainly enable prime breeding. It has to take into account that we are dealing with emerging phenotypes. Again, these emergent phenotypes, they are coming and they're generated through the interplay between the different traits, the different gene networks, and whatnot. And to make things a bit more complicated, the patterns of G by E are not only dependent on where we are positioned in this environment gradient, but it depends on where we are going to, where we are intended to go, what are the traits that really matter, and how the environments are changing. So, for example, you take this environment 1 and 2. On the lower x-axis is about 600 grams per square meter. This yellow area is one trait that explains most of the variation. So if we compare two genotypes in these two environments, the one with the favorable alleles is going to win regardless where we are. So in this particular case, if you compare these two hybrids, one with better silk elongation, for example, relative to the one that doesn't, regardless of the environment, there's not going to be G by E. But let's move this environment 1 to environment 2 closer to the 1200 gram per square meter. That's kind of central Midwest, probably is in some good environments in Argentina. Let's keep focusing on this silk elongation rate and pollination. What we are going to see is a genotype by environment interaction, yet this interaction will be due to a heterogeneity of variances, whereby at the 600 yield gram per square meter environment, the genotype with favorable alleles or traits is going to yield way more than the unfavorable one. But at 1200 millimeters, you can see a gram per square meter, you can see the yield variation explained by this trait is very little. Therefore, this combination of genes and traits is not going to make a big difference between these two hybrids. So now go to to an extreme. This environment 2 is now in 1900. This is a potential environment fully irrigated. And think about transpiration, and then you start looking at high productivity requires carbon fixation, therefore it requires water. In this particular scenario, the trait that gives the highest, best adaptation is this canopy size or photosynthesis transformation, or RUE, if you will, and it's going to trade a lot of water for a lot of carbon. So now, if you compare the silk emergent trait with the canopy trait, the silk emergence trait is going to give you heterogeneous variances, but the canopy size and the water use is going to give you this lack of correlation type, and now the rankings of your hybrid is going to change. And if we go, an extreme, we have the combination of both types, and that's where things can get really ugly for a breeder that need to start defining where are going to do the selections. So in the end, what we need is a new type of biostatistical machinery to predict these genotype by environment interactions. And May alluded to machine learning. And yes, statistical learning has a tremendous power to learn from data and the patterns that are embedded in those data sets, but it's not going to take us where we need to go, particularly as we think about making long-term predictions for carbon sequestration, water use, and nitrogen, if you will. If we don't harness the scientific understanding of 100 years of crop physiology, agronomy, uh, and agronomy science. So what we are advocating here is a method that combines both the symbolic AI, artificial intelligence, which encapsulates scientific understanding, with the sub-symbolic, which is statistical learning, and the, if you will, the deep learning. Um, we are not going to deliver the solutions that the breeders are going to need now into the future. So let's start talking about this AI or symbolic AI, and think about the phenotype as a function F that it's determined by genotype management and environment. And here we can partition that function in things that are detected or determined and things that are not. So for instance, take this function Q, which is detected, can be photosynthesis, transpiration, essentially is what we know. So Q can be a set of crop growth models that may mentioned before, and essentially this is a formal representation of what we know, and more important is a cognitive model of how processes, elements are causally related to each other. This is really fundamental because the networks in deep learning are not. So going back to my argument about symbolic and sub-symbolic AI integration, these cognitive models can put many restrictions and constraints to what the deep learning model can search in the state space. So now we can think about the crop growth model then. So in a set of parameters that we know in the crop growth model, and set of parameters that we want to estimate, and of course a number of inputs and environment, which is going to be used to predict the genotype by environment by management interactions. So how we are going to integrate these two? First, think about the crop growth model as a data generation function of a vector of known parameters, and of course the ones we want to estimate, and the environment. And the sub-symbolic model here I use in the classes here at University of Florida. This is a Henderson model where it should be familiar to the breeders, so you can utilize this type of genomic prediction models to apply to different physiological traits. For example, Y could be RUE, so we can have a function G that will provide a mean mu and a vector of marker effects. And we can combine this in the likelihood function that it should be very familiar to those of you working in plant breeding using Bayesian approaches. So now we can have these dynamical networks with the genomic selection, and it's a very transparent integration of this symbolic AI, which encapsulates years of crop science with the number of years statistics development and of course learning. So is this approach, does this approach work? And the answer is yes. We published two papers recently, one in plant physiology with Christine Dippenbrook, working at the Corteva, and this data set includes 2,367 hybrids in many locations. Each of those points is an experiment in a wide range of evapotranspiration environmental gradient. So in places where the distance between the training set and the prediction set, let's say well water environment, high evapotranspiration, as I showed you before in the G by E theoretical argument I made, the distance is not large, therefore the predictions are not, the difference or the accuracy or the improvement between these AI symbolic, sub-symbolic relative to a genomic prediction, GBLAB, for example, is not great. Actually, it's pretty much close to zero. But as you start moving into the drought environment, lower evapotranspiration, so this is a drought stress where the distance between training and evaluation includes sets that the genotype by environment interactions are important. You can see that having these functions that encapsulate the ability to predict G by E start playing around a significant role, and therefore the accuracy improves significantly. In many cases is double the what you can attain with GBLUP. And in the second paper, we expanded this to 7,000, more than 7,000 hybrids in about 6 years of the drought program. And you can see the same story. You can compare the accuracy on the x-axis of the whole genome prediction, that was a base A prediction, relative to the accuracy of a crop model combined with the genomic prediction, as I showed you before. And you can see in many cases you have 0.2 as kind of an average, and this by adding the functionality of a crop growth model, you can increase that by 30% or more. And that is for a complex trait like yield and flowering time, whether it's shedding or silking and similar things with kernels per year. So the approach, it has some limitations, as the distance between the environments are too close, so that's not giving you a whole lot. But when the distance between where we are standing today and where we're going to go in the future, in future climates, and particularly as you think about higher temperatures and drought, that these type of methodologies can become critical tools for the breeder. And the other thing is, because the training occurs at the agronomic and physiological level, we have genetic predictors combined with environmental predictors that then we can simulate for many different locations and environments across this evapotranspiration gradient, the yields that can be attained by different hybrids. And in this particular case, the breeder can define their target population of environments as part of the distribution within the evapotranspiration range, and you can see what you can make a decision there on what hybrids you want to choose. But also the breeders can start interacting with policymakers and decide how they're going to produce food, sequester carbon, and utilize water in the best manner. So you can start thinking about these multidimensional breeding selections, depending on where they are in that gradient, the source of water. For example, think about fixing carbon or do carbon sequestration using irrigation. It's absolutely nonsense. Again, we can start coming together as groups that can start having dialogues to evaluate how we are going to breed in multiple dimensions for today and into the future. And here I want to transition a little bit and sort of discuss this paper that claimed that not genetics underpin recent male gains. And the beauty of science and the methods and the data that we have enable us to test some of these statements. So the first thing we would like to get back to is the breeding process, the same data as I show you in the first figure, but now display in a different way. So over the 100 years of plant breeding that occur in the United States, if you look at the regional anomalies and differences, essentially from the 1900 to 2000 mean, you can see precipitation went up in that central corn belt. The minimum temperature went up and the amplitude temperature went down because the maximum temperatures did not change that much in the same period of time. So these are significant trends. The deltas are important, and I will show you later. So we have a concurrent change in the climate as breeders were making genetic progress all throughout this time. So to test a little bit of that statement that was published in the National Academy of Sciences in the United States, the experiment we would like to run is to set the genetics constant. So we take a subset of hybrids commercialized between 1930 and 1990, which are the dots, the black dots, in the figure on the bottom. So each point indicates along this gradient of environment when it was commercialized. So with that, so we have a set of hybrids commercialized over a common period of time. So we can calculate genetic gains. And the next thing we want to do is look at experiments that were conducted in a period of time that came after that. So it's about 30 years. Again, this is data published by Dewey, Cooper, and in the past. So we can interrogate these data sets now using a different perspective. So now we have a matrix of environment. So in each year, we have all the genotypes from 1930 to 1990 represented. And of course, we have also the management, considering the agronomic dimension in this PNAS paper, normal density, low and high. So now we can add the agronomy. You can separate genotype, management, and environment. I show you the environment is changing, the genotypes were changing, and the density agronomy has been changing. So now we have all the factors, so we can calculate the genetic gains for every year at each of these three managements. So we can look at the grain yield and experiment, the year of commercialization. Remember, this is the time, but is the commercialization of the genotype, not the year that when the experiment was conducted. So take this is one graph out of many that can be constructed. So the slope is the genetic gain. So now we can go back and ask the question whether these genetic gains were zero, as he claimed in the PNS paper, or genetics were significantly higher. And what you can see here in the low densities, we can call it this is a pure G contributions. The genetic gain, again, the slope of that relationship between grain yield and year of commercialization was about 2.5, pretty much constant throughout 20 years, from 1990s to almost the 2020 time. If we grow the same experiment at normal densities, which is the grower's density, we can see a genotype by management interaction. Of course, the management interaction can, because this rate of genetic gain are different, and almost double. So essentially, what we can see here is that at the normal density, the rate of genetic gain is about 7. Well, actually, it's more than double. My apologies for that. And essentially is reviewing the statement on the PNS paper. And if now we look at the high plant populations, we can see that is even an acceleration of the genetic gain, with the rate of genetic gain in the last years are higher than the previous year. So it's not just environment, it's not just agronomy, it's genetics that is magnified their effect by the management and the environment. Again, think about the last years. If you take that filter red line is about 12 grams per square meter, is about 3 to 4 times the rate of genetic gain and the low plant population. So again, I think if we make a single statement and compare the rate of genetic gain were always positive over 30 years of experimentation in Central Corn Belt, which bring us to a point what they were trying to say in this paper in terms of genetic gain for yield. So the number of hypotheses that we can formulate to interpret this, and one is genetic gain increase because of the favorable water conditions, which is one of the variables that change. The other one is genetic gain increase because the long-term adaptation to lower amplitude temperatures. So essentially, the environments were changing, and we were adapting to that. Because of the environment change was slow, we were just shifting across that environmental gradient, as I showed you before, in a very coherent manner. Essentially, there was enough time for the breeding program to adapt to that slow change. So test the first hypothesis. What we know from corn is that if we impose water deficits the yields are going to go down regardless of these are double crosses or single crosses. So here is one example in terms of yield. You can see the red bars are double crosses, hybrids commercialized between 30 and 60s, and the delta is a little bit higher than the white bars, which are the single crosses. This is due to increased barrenness, scatter grains, and kernels per year. So in this particular case, and it can be confounded because the environment and the genetic gain are both going in the same direction. So again, maybe the PNS paper has something that we didn't know. So we can go back and use some of the simulation studies we conducted to ask the question, is this impact or increase in rainfall yield is enough to explain the changes or the difference in rate of genetic gain? And the answer is absolutely no. If the rainfall precipitation anomaly is about 100 millimeters, we go from about 500 to 600 millimeters of evapotranspiration. And at the best case, we are talking about 1 ton of yield gain over 100 years, which is just a fraction of what happened. So we can test this environment hypothesis, and we can cross it out with based on a quite a solid simulation and experimental studies. Next one I think is more interesting is this co-adaptation of the germplasm as the environment was changing over time. And one of the traits that are affected by the temperature is the radiation use efficiency. We conduct experiments over 5 years, and you can see there's a consistent improvement in light use efficiency, about 0.3% a year, which is about 30% in 100 years. If you think about light use efficiency, pretty much is a linear multiplier to light interception and mass. There's a fair, it's a 1/3 of the yield can be explained by this trait alone. And the other thing we know is the amplitude temperature is going down, and the amplitude temperature is highly correlated with the reduction in light use efficiency. The lower the amplitude, the lower the light use efficiency. So in this particular case, the environment was working against the genetic progress. So essentially, think about these lines moving in parallel up, while the environment was just pushing downwards. So over 100 years of breeding, essentially breeders had been using genetics to counter out the effect of these negative forces that are reducing the light use efficiency. So the interesting thing here is that we need a framework to start thinking how we are going to breed for climate change. And think about if this slow change hypothesis on amplitude temperature holds, perhaps we don't need to do anything. But if, as we expose our germplasm to new environment and genotype by environment interactions may start coming in the form of lack of correlation, what we can see here in this graph is that in the US, yes, the sampling environments correlate with the target population of environment. Essentially, those two things were moving together. So the covariance was high, therefore improvement was coherent between the two. But if you think about the multi-environment trials in the 220 would not match the target population of 2050. Again, maybe the form of more well water going into the drought environment or heat environment, we can't really get a continual count that breeding is going to deliver the solutions as we hope. And we cannot move it too fast because it's going to reduce the covariance between the current and future environments, the performance. So if we go too fast, we starve today's populations. You go too slow, we starve the future population. So we need a system not only to monitor where we are at, but how we are making progress and how the multi-environment trials are tracking with the target population of environments of 2050s and so on. So in summary, I would like to leave more with a message than what I talk about. I think we can reimagine agriculture as a solution to climate change and human health problems. I think we need to advance predictive breeding in this combination of artificial intelligence, symbolic and sub-symbolic, to enable the circular bioeconomy. And as I alluded and tried to go show you in the last slide, we definitely need a framework for plant breeding for climate change to inform policy and decisions. It's not just enough to put a statement that cultivars would be an option to fight climate change. And with that, I would like to